Hello, everyone. Welcome to Foresight's biotech group. Today, we're really lucky to have Peter Lidsky here with us. He studied at University of Moscow and University of Zurich, where he was working on virology. And now he's at University of Hong Kong now, and he has developed a pathogen control hypothesis of aging. Without further ado, Peter, thanks so much for joining us, and we're excited for your presentation.
Okay, so hello everyone. Thank you for coming. So I want to discuss why do we age and our progress on the search for the paradigm. So first of all, aging is like a risk factor for many diseases, so-called age-related diseases. You see that cardiovascular, Alzheimer's, cancer all goes up with as we age. And so it seems to be, it should be in a focus of public health. However, if you look at funding, the funding is not great. The National Institute of Aging has 9% of the budget. However, if we look where this money goes, we find that only a tiny proportion of this money are going towards fundamental aging research. Most goes into Alzheimer. Why is it? Moreover, this was a paper published recently by Gerald Shansky. He said that we already exhausted our opportunity to extend human lifespan. So human life expectancy was growing the past century. And now we seem to be on the plateau. And if we want to grow, we need some breakthrough in medical sciences. So Eric Worden, who is one of the key people in the aging field, reacted to that this study should give pause and ponder enthusiasm on life extension, and we should focus instead on the task at hand, improving health span. What is health span? Instead of lifespan, we want to focus on health span. The same message came from the director of the huge philanthropic organization, Evolution, Mahmoud Khan, who also said that my father used to say, I want to walk to my own grave. So we want to stay very healthy until we die, and then die very sharply. So it seems to be that instead of focusing on this underlying reason, we need to address all these exponentially growing curves individually. And these are only four. There is also obviously arthritis and kidney disorders that are not depicted on this slide. And it seems to be a really strange idea, because as I would propose to address cough, temperature, and headache to cure COVID, it will clearly not work. So why educated and intelligent people propose switching from lifespan research to health span research? Because, surprisingly, we don't know what aging is. So this is a paper that has been published last month by a huge group of authors led by Vadim Gladyshev. He did a survey between experts in the field, and the conclusion is that everybody, to the very basic question, what is aging, give different answers. They could produce, how you say it, word clouds, right? And what is the most important unanswered question in the field is aging. The only hit here. So we just don't have a good definition. The field doesn't have paradigm, and this is a critical bottleneck that I believe limits us in our attempt to fight it. So why it's so important to find paradigm for aging? For example, the very first question we will ask ourselves when we have a paradigm, is aging a damage accumulation process, some type of an entropy, or it is a programmed genetic process? In the first case, we need to help these dots to move back to their original places. And in the second case, we need to find this gun and remove the bullets. So these are two completely different projects. In first case, we need to fix something. In the second case, we need to break something down. If we take a wrong fork at this moment, 99% of effort will be probably lost. And most of the researchers took the fork towards anthropic models, this damage accumulation, and I believe it's wrong. So another critical question that we have is it possible to rejuvenate the human body? Earlier last year, we have run a debate at the Foresight Institute. So Alison is smiling. So it was a real tie. So these two guys is it possible to to rejuvenate people? And this is a critical practical question, because in one case, we need to take old mice and try to rejuvenate them. In other case, we need to take mid-aged mice and try to freeze them in their aging. And we still don't. So the formerly guy who was saying rejuvenation is impossible won, but it was a tie. So the votes of jury were very close to each other. And I don't think in the next debate, in a couple of years, the victory will still be with Peter. I am trying to answer this critical question, what is aging? And my approach is the easy questions first. The first easy question is, why do different animals age with different speed? If we take a rat, it lives 2 years, maximum 4. Squirrel, which is same size, same diet, can live for 20. So very similar animals, related, tails are a little bit different. Why are they so different? Traditional take is that their efficiency of damage control is adjusted to particular ecological niche, and the factors that affect these parameters of ecological niche are extrinsic mortality, maturation time, metabolic rates, something very, very simple, understood, and boring. But this is not true, because if you look at animals that can live longer, so these are like all mice-size animals. Brand's bat can live for 40 years, sparrow can live for 20, frog can live for 25. So these mature very fast. These have high metabolic rates. These two have high extrinsic mortality. They are clearly outliers, and the factors that are typically listed by gerontologists they clearly have exceptions that they cannot explain. And squirrel and rat is only part of a gradient. Or there is also this naked mole rat that practically doesn't age. So if you look in this picture, so we see that mortality doesn't grow, and still we have no explanation why it happens. On the other side of the gradient, we have this northern quoll. These are marsupials living in Australia that die very, very soon after the first reproduction. This is called semilparity, and it's also can be found in Pacific salmon. So a fish going up the river lay eggs and die in some strange form of death. These northern quolls, they also have a hormonal spike that is a mechanistic cause of their death. Surprisingly, this semilparity evolved several times on the tree of these carnivorous marsupials, but not in rodents, not in insectivores, never in carnivores that occupy similar ecological niches. What happened? Why does it happen only in marsupials? It is not clear as well. And looking at this rainbow, where are damages in this equation? Where is entropy? Okay, if we look at aging body, we see the damages, but we also see damages in any other diseases. In COVID, we also see many damages. Does anybody propose to cure COVID based on classification of all the damages and trying to deal with them one by one? I think this is not the way to go. And so my approach to conceptualize and rationalize the observations is evolution. And this is a classic approach that is used in biology. Nothing in biology makes sense except in the light of evolution. Only true. So I now just remind you what modern evolutionary concepts on aging. So these two guys, they propose so-called selection shadow concept. Idea is that animals are dying of predation, hunger, cold, and other extrinsic reasons that are not dependent on aging. And by a certain age, no animals or very few animals are surviving to the age X. And this makes aging neutral. It doesn't matter what happened to the animal after it is dead with 100% probability. But this model predicts that animals should not age in the wild. And this is not true. If we look on mortality in lions, let's say we immediately find that 5-year-old lions have much lower probability of dying than 14-year-old lions. And lions survive to the age of 14 in the wild. So if potentially there would be a mutant lion in which this curve is a little bit more flat, this lion would live longer, leave more progeny, and this variant would be selected by evolution. But by which reason it doesn't happen is not explained by this model. So the person who tried to explain that was George Williams, who proposed his antagonistic Pleiotropy hypothesis. He hypothesized the existence of genes which are beneficial in the youth and detrimental at the old age. And if these genes exist, then they help a huge proportion of animals that are still there, but are harming only a small proportion of animals that survive to age X. And if we believe the existence of such genes, then indeed it can explain aging. However, the problem with this hypothesis is that nobody yet shown a single pleiotropic gene. There are genes with the two functions, and one can be beneficial, one can be detrimental. However, this is not enough to explain aging. These two traits must be evolutionary stable, linked with evolutionary stable link. And the evolutionary stability of this connection has never been shown. And I tried to imagine this even theoretically, it's not possible. I didn't came up with a single example. All the examples that were proposed by the members of community clearly are not good, and I can clearly see how evolution could disconnect them. So the next stage of progress of the thought of the ideas has been done by Thomas Kirkwood. It's disposable soma theory. He proposed the existence of fundamental trade-off between longevity and reproduction. And he proposed that energy is the main currency that mediates this trade-off. So we have a limited number of food in our life, so we can invest it either in reproduction, either in repair. We overinvest in reproduction at expense of repair, and we just produce a lot of progeny. And these fast reproducers, as in this picture, they can overgrow the slow reproducers. However, this model has mathematical problems I don't want to discuss right now, but it also has biological problems that are easier to explain. For example, if we feed animals that they have enough energy for reproduction and repair, they get fat and they live shorter. They don't live longer. Males age faster than females. Males invest much less in reproduction. Full consumption decreases with age, should increase according to the prediction of the model. Poor ecosystems like Arctic caves, deep water are all enriched with long-lived species. Longevity seems to be much better saving strategy than over reproduction. And all the entropic models that are people now using, in the end, their pedigree is coming from these two very questionable models. We exercise, and these models are specifically bad in explaining natural observations, variability between species. Everything what flies lives very long. Also, there is a huge difference between longevity in case of eusocial insects. So the termite queens can live 50 times longer than workers, while they have the same genome. Why it happens? Mneic mole rats, immortal, not clear why. These two species are simiparous, not clear why. And this is not explained by the classic model to me. Also, they exclude rejuvenation because they both consider that aging is bad. And if aging is bad, then if rejuvenation would be possible, it would be likely invented by evolution, at least in some cases, where let's say, an old animal found a lot of food or some very good conditions, then it should rejuvenate, and this should be beneficial according to these models. Since we don't see rejuvenation in nature, probably it's mechanistically impossible because evolution couldn't invent it. So, and then it's questionable if we can produce rejuvenation with our biotech. However, I don't believe these models. It's a good news. And we exercise this programmed aging idea, and the two key questions that have been asked were considered to be critical. Why aging cannot be programmed? Question number one, it's not clear what this program is good for. Idea of life is reproduction. If I kill myself, I cannot reproduce. It's clear minus. And second, why don't we see mortal mutants? If we treat our flies or worms with mutagens, we get flies without eyes, without wings. We never get immortal ones. So if aging is a program, it should be in our genes. If it is in our genes, we can be able to break it down with the mutation. Never happens, so aging is not problem. I have built a model that can address both these questions. First of all, start with first. What the program can be good for. Are there accepted examples of adaptive death? Yes, there are. So this is so-called abortive infection in bacteria. If infected with a bacteriophage, bacteria is probably committed to death anyway because it cannot get rid of the pathogen. And it tries to kill itself to prevent replication of the phage and infection of neighboring bacteria, which are probably its relatives. And this is a classic part of the immune system. Phage is trying to inhibit it. Bacteria is trying to overcome this phage inhibition, invent other ways to kill itself. It's all very well described. So can something like that can be imaginable in our population? Yes, in principle it can. So if we have a parasite and a host, so this is an example of fish and its parasite, and the fish cannot clear the parasite, let's say. So then the probability of being infected increases with the age. So you see that fish of the age of 8, 100% probability are infected. Now, imagine this parasite doesn't sterilize fish, but let's say parasite sterilizes fish that it cannot reproduce. Then, after a certain age, everyone will be not able to produce any progeny, zero cost for zero value for evolution, but this fish can still infect their relatives that are likely swimming somewhere nearby. And in this case, these fish are of negative value. They are detrimental for their own genes, right? And then for evolution, it can make sense to kill all these fishes just based on age prophylactically. And for this, the key parameter in this model is this relatedness in the in the networks of interactions, because individuals should infect their relatives. For that, we developed this model. We published it a while ago. So the model of territorial animals that crawl on the surface, and this somehow recapitulates what happens in nature. This like GPS tracked wolves in Minnesota. The idea is that viscosity of the population structure, it can preserve relatedness in the interaction between individuals and ensure that pathogens are spread from brothers to brothers. And so this in this model, so we have run the epidemics. So if we run epidemics of the chronic sterilizing disease in short-lived individuals, then we see that epidemics is not that strong. If the individuals are long-lived, then the pathogen is very happy because these infected individuals are living for longer time. And if we introduce a long-lived mutant in a short-lived population, then of course, without pathogen, it takes all the population because it lives longer, produces more progeny, and is very happy. However, what happens if we put a long-lived mutant in the presence of the pathogen? So in this case, these pathogens, they can prevent this long-lived mutant from taking over the population. So you see that epidemics in long-lived part of the field is stronger, and animals are sterilized to a higher extent, and they are displaced by population pressures from these neighboring non-infected fields. And this is, in our opinion, the reason why the biological function of aging, to remove old and potentially infected animals. Now we are why we don't see immortal mutants. First of all, we see what we can consider a gain of function mutant, accelerated aging, but we never see an immortal Voldemort. Why? First of all, the question is incorrect. We see immortal mutants. Naked mole rat has a common ancestor with squirrel and a rat. And so along this branch, at some point, like mortal mother should have produced an immortal pup. But it clearly happens very rarely. And it seems to be that aging is somehow fixed by evolution. Why can it be fixed? To answer this question, we need to look at species where it is not. And these are eusocial insects. Eusocial insects have this big discrepancy between queen aging and worker aging. They clearly have two programs. And in these animals, it was shown that pathogens, when infecting workers, can switch their program to the long life, because pathogens don't want us to die. They want to spread. And for that they need time. And paradoxically, pathogens in our fight against aging are our allies because they want their host to survive. And just read this figure. These are how workers die. These, I think, black lines, how the queens die, and yellow line is infected workers. Idea is that evolution developed some security belts that prevent pathogens from manipulating and switching off our aging. And these security belts also prevent mutations, also prevent medicines, unfortunately for us. In principle, phenotypically, when we run into one of these security belts, we can interpret it as evidence for pleiotropic gene. So this is the first and the only model that can answer both questions in the context of programmed aging. And this is a big claim because so this is probably many of these hallmarks of aging, a catalog of all the aging-related phenomena that people often cite as a paradigm. I believe it's a catalog, and we believe that immune aspect of aging is key, and generally that gerontology should become a part of immunology. This is a very big claim. So because let's say if I go to the conference next time, last time the aging was in the section of physiology and metabolism, I believe it needs to be with the immunity infection. And of course, people don't believe me because this is the claim is too big. And what we are doing right now, I believe the most important part of the project is to test the evolutionary theory. This is my family doctor. He was saying that the diagnosis should go first and only then the treatment should go. And we are working on formulating the ecological predictions that actually our model on verbally, it can explain things much better than any other model. You can read our review, but now I have no time to go one by one. So I can tell you about two of the ideas that we have that are crazy enough to be mentioned. And the second part is more fundable, more close to the drugs. So we believe that senescent cells are part of antiviral immunity. And this is something that we are like exercising as a fundable project. But also it is fun. So first of all, how do we explain why mole rats live so long? They are eusocial. Mole rats have queens and workers like bees and ants. And this population structure, according to our model, can bring back this bacterial type of defense. So if the very first mole rat infected with any pathogen kills itself, this is a harakiri knife, then the colony can be clear of any pathogens. And this is much more efficient way how to control pathogens than aging. Then aging can become redundant. And indeed, there are a couple case reports that indicate that mole rats are hypersensitive to infection. So if they are infected with some viruses, they die immediately. This herpes simplex is a virus that mice actually do not care at all. So the idea is that they evolved eusociality. This stimulated this suicide in response to infection, and this led to an evolution of negligible senescence. Second project, we are trying to explain why similarity evolved in these carnivorous marsupials and not in other mammals. And idea is that the model predicts that these animals should have some unique disease that is not around in carnivores or insectivores that have similar population structures, similar ecology. And indeed, we have idea what disease can it be. So you may have heard about devil facial tumor disease that is ravaging. This is a transmissible cancer that emerged like a couple decades ago in a female devil, and these cells are transmitted from animal to animal. And this disease has very unique features because it's highly transmissible, highly lethal, and has a long incubation period. And according to our preliminary data, it can indeed stimulate the evolution of similarity because these animals are replicating annually, and the peak of the infection happens during breeding season. And typically, the male, if infected during this breeding season, has no chance to survive to the next breeding season. But it can go around and spread infection during the non-breeding seasons. And then there is not much evolutionary reason for this mammal to survive. And while devils now are not semiparos, but evolution of similarity in devils in response to this DFTD has been already discussed in the literature in the context of disposable soma, but we believe this is disease control. Funny enough, there are now two independent DFTD lines persisting in devils, showing that these mammals can indeed be very susceptible to facial tumors. and our physiological model of what happens. So we work on epidemiological model, but physiological idea is that these animals have some specific embryogenesis. They have cooperative and they have inflammation between embryo and mother during embryogenesis. And this may create a trade-off between non-self recognition and feeding the embryo. Idea is that this feature led to this embryogenesis thing led to the poor non-self recognition, which is also published in a small case report. It's a very small study, but it also indicates our direction. And this leads to the presence, spontaneous induction, repeated induction of transmissible tumors that results in the evolution of similarity. So two crazy predictions that we want to test after we finish the theories is hypersensitivity of naked mole rats and presence of transmissible cancers in other marsupials, other carnivorous marsupials. Devils are probably the largest, and that's why we see the transmissible cancers there first. And now coming to the cellular senescence stuff, I still have 9 minutes. Excellent. You may know that cellular senescence is when cells stop dividing, but they don't die. They're resistant to death. They don't resistant to apoptosis, and they also produce a lot of signaling molecules. First of all, inflammatory. That can make other cells secondary senescent, and they generally create inflammation. And this accumulation of these senescent cells lead to metabolic disorders and cancer, and it exacerbates many other diseases. But the biological function of cellular senescence is not clear. This screenshot has been taken from the web page of the organization, International Cell Senescence Association, that I also a member of. And what is cellular senescence? Cellular senescence is a programmed state of stable cell cycle arrest that is accompanied by a complex phenotype. Okay, this is observation. Senescent cells play a role in physiological processes such as tumor suppression, wound healing, and embryonic development, because paradoxically, they can contribute to aging, cancer, and age-related disease. So this is, I think this is not clear. So clear that nothing is clear. That because you see, cancer is mentioned twice in a positive and negative way. And so I don't understand fully the biological role of senescent cells from this statement. In 2016, there was a small paper that proposed that cellular senescence is an antiviral defense mechanism. This is a good paper. I encourage all of you to read it. It has some disadvantages that probably did not make it to be widely accepted. So in making a long story short, we repeated their results. So these are control A549 cells. These are senescent A549 cells. We infect them with GFP-labeled viruses. We infect them, and you see the difference is pretty clear. We can try it with different inductions of chemical inductions of senescence. They all get antiviral. Again, it's pan-virus effect. So you see here 5 different viruses belonging to 4 different families. All of these senescent cells can. So the thing on the Y is amount of virus produced 48 hours after infection. You can see that all these senescent cells have reduced amount of virus produced, but by some reason, this induction is not as antiviral as this and this. Okay, and we decided to use it to find the mechanism. We did RNA-seq on just control cells without viruses, and we found like 16,000 differentially expressed transcripts. Best luck to find anything in this soup. But because cellular senescence changes a lot in cell physiology. But if we focus only on targets that are activated in the antiviral probes in this and in this, but not in this, then we boil it down to less than 300 differentially expressed targets, and they are all enriched with the interferon-induced genes, immune system, most of them, and we tested if these interferons are indeed responsible for the antiviral function of senescence for this phenotype. So we found that yes, it is. So if we knock out, so this has been done with the GFP-labeled virus again. So these are control cells, these are senescent cells. Here we tested knockouts. If we knock out IFNAR, which is interferon-1 receptor, then the senescent cells can accumulate some green. Same happens if we knock out STAT1, which is just downstream from IFNAR. also here, and the just senescent, wild-type senescent cells are here, so it's a strong effect. Same was true if we use the inhibitor of this JAK-STAT pathway, the same actually MAC pathway that STAT1 is involved in. So it seems to be that these cells just produce interferons and activate antiviral signals in them, and they can also can protect healthy neighboring cells. And we can also show that if we take supernatants from the senescent cells, we put them on healthy cells, and then we see that virus reproduction is reduced. So a very basic result. It was already proposed in the previous papers, so it's not entirely new. We just bring in some mystic aspects. And the big model that we have right now is, I call it immune militia model of cellular senescence. This is my branding of the idea that was proposed before me. So idea is that viruses can induce cellular senescence. These senescent cells make these cytokines. This is called senescence-associated secretory phenotype. These cytokines protect healthy cells, communicate with the immune cells, and mediate virus clearance. After virus clearance, the senescent cells that can still contain some virus, if they would go back to non-senescent state, they need to be dead. If they are not killed, then they start to make problems. So why militia? I believe that these cells can be recruited to be defenders against viruses from any other job. Then they do this defense. They can communicate with the professional soldiers, which are immune cells. And after the war is over, they must be demobilized. If they are not demobilized, they start looting. This is the mnemonic paradigm, mnemonic hypothesis. With this, I want to thank people who were involved in this project before at UCSF. This is my ex-boss, and here are two genius students that were working with me. And here is my new lab, and I'm very happy to take all the questions.
Thank you so much. That was totally fascinating. I don't see any questions in the chat, but if you guys want to raise your hands, we can move through your questions that way. Okay, Vadim.
Hello.
Hi.
Thank you. It's a great presentation. I just wanted to first discuss the basics. So meaning that programmed aging theory versus non-programmed. To me, you seem to dismiss a large body of data on molecular patterns of longevity and also damage. So for example, we literally see mutations, they accumulate with age, epimutations, modifications long-lived proteins, telomere shortening, oxidative damage. We literally see all of them, which will grow with age. You dismiss this based on differences in longevity across species. But we also see patterns across species. For example, transcriptomics, epigenetic, there are many papers where we could explain why different species have different lifespan. On the other hand, you say that aging is programmed based on one example, literally, of the naked mole rat not showing increased mortality with age. So here, to me, you mix up mortality and aging because the organism may age, but might not show increased chance of dying with age. Because mortality is caused not just by aging, but for example, by infections or accidents, other features. We don't know why naked mole rats don't show increased chance of dying with age, but they do age, and it's very clear based on transcriptomic patterns, epigenetic patterns, and other features. So to me, there is no evidence that aging is programmed. You say that there are no mutants detected, which is true. For example, in humans, we have mutations every single gene, but we never have a situation when somebody stops aging. And this is true for any individuals in any species. There are so many examples of such mutants. We've never observed it when the program is disrupted and organisms stopped aging. To me, you dismiss something which we clearly see and introduce something which we never see.
Yeah, thank you for this question. It was a long one, but I will try my best to answer it. First of all, you say that you see damage. You have no evidence that damage is cause of aging. Because if you look at any other disease, any infection, any diabetes, whatever, you will also see a lot of damage. And you also can build clocks there, how it develops with time. And I think even in diabetics, telomeres are getting shorter, I think, faster than in non-diabetics. But nobody connected these two. So there can be some causation, but it can be much more complex than. So you clearly don't know what is the primary reason, what is the secondary reason. So to me, there is no scientific reasons to believe that aging is caused by damage. The models, the data can be explained by different models. Second, you said that I dismissed the damage, the non-programmed theories not based on only mole rats. I dismissed them based on analytical argument. Mole rat is just an example showing that longer lifespan or almost non-aging has evolved to some extent. I think in this discussion, in the end, will boil down into the definition of what we think non-aging is. Mole rats do not show actuarial senescence. The mortality doesn't grow with age. Ecologically, is it non-aging or not? Then you say, okay, they have some biochemical thing, but is this biochemical change evolutionarily relevant? So we don't have this answer. So in any case, I think the example of mole rat is quite fascinating. And if we would make a mouse or a human with similar parameters of aging, it would be a big success. Yeah, hope I have answered a big part of your question, right? No? Sorry about that.
I suppose we can move on to the next one. How about let's call on Chris.
Okay, thank you. A very intriguing talk. I'm not an expert leading in the field like Vadim. I just want to hear more of your critique of antagonistic pleiotropy. For me, the papers that really made me understand aging are Medawar, I think it was Evolutionary Theory of Aging, 1966, Brian Charlesworth, Aging in Age-Structured Populations, that there is a fundamental trade-off with the lack of selective pressure post-reproductive years versus pre-reproductive years. Post-reproductive years, you've got no passing on of the genes that got you there. And antagonistic pleiotropy in a broad sense. So you have good genes that help you get to reproductive years. They go bad. There's nothing that prevents them from going bad post-reproductive peak. You've got bad genes that are silenced until reproductive years, and post-reproductive peak, those can be unsilenced. And I don't see a conflict with reversing aging by silencing those genes, reversing the entropic, basically, changes that occur.
Yeah,
But
so
again, I'm not an expert.
I
Go
see
ahead,
your question. I think I will try to answer it as well. So the answer would be that you now divide aging into reproductive and non-reproductive, and you assume that reproductive aging happens somehow by itself, and then you derive the somatic non-reproductive aging from it, right? But reproductive aging is also part of aging and probably a reason of consequence. I'm sleepy. Consequence of the main aging. So there is no reason why reproduction should go down with time. We have the whole aging that includes reproductive aging and non-reproductive. People divide it into two pieces. Declare that reproductive aging pre-exists, and then the pre-existence of reproductive aging explains the lack of selection for aging and death of the rest of the body. Did I answer your question?
Part of it. I think I agree with part of what you're saying. Take an example of the mouse versus the mouse with wings, right? So the mouse lives 24 months or whatever, the mouse with wings, the bat flies away from its predators and can reproduce much later, lives 20 or 30 years. So there's a evolutionary force that would give the bat reproductive ability and sexual selectivity preference longer than the mouse.
Yeah, so the effects of extrinsic mortality on aging is a ground of huge debate. You can go to my Twitter we discussed, but it is very specific. So people don't believe it's there. There is no direct effect. It can be indirect through changing the reproduction schedules, but there is no direct effect of extrinsic mortality. Moreover, in other mouse with wings, like a sparrow, has very high mortality. So 50% of sparrows dies a year, but they still live very long. And they have more regular mouse type lifespan in the wild, and they still produce a lot of eggs and for a very long time. So there is it's clearly a counterexample, and I don't see any rational idea why reproductive aging should go with time down. Okay, if let's say I'm an animal, 99% of all others of my age died. So why should I die? Why is it a good evolutionary strategy? Other way around, I should use this opportunity to reproduce myself. And this has been formalized mathematically. I'm not sure I have a slide in this section, but you can definitely go to my Twitter or you can email me. No, I don't have. Daniel Promislov would be the right person to bug. Also, Hal Caswell, they did math on this. So also you can resort to works of David Resnick, who showed that in guppy, higher mortality resulted in slower aging. So not longer lifespan, but slower aging in terms of physiology. If you email me, I can send you the references. I'm not sure if I gave you a comprehensive answer, but I tried my best at least. Okay.
Comprehensive and partially convincing. Thank you.
Great. Thank you. Next up, we have Moritz.
Thank you for the talk, Peter. Really appreciate it. To me, it's also very clear that aging is adaptive. I find it such a big feature of life, and that evolution hasn't optimized for that. It seems crazy. So I'm very convinced that it's adaptive. The best sort of the simplest answer that I've read is this paper. It's 2011 paper. I was just looking it up. It's called Change in Aging Senescence as an Adaptation. And basically what the author did, he modeled several species. And the idea was just, I have the same species in a changing environment. One dies, the other one doesn't.
Mm-hmm.
And it turned out, just like over thousands of simulations, the aging species always wins. Why?
Mm-hmm. Mm-hmm.
Because over time, it is just better fit to the changing environment. So basically, changing environment, if you die, you're better fit to the environment than your non-aging, your non-dying, the same species that's not dying. And it makes perfect sense to me. And I find this is a simpler argument than what you're proposing, which is involving the pathogens. It might be, I find that the pathogens are downstream of that. It makes perfect sense to me. But why involve the pathogens in this model?
Yeah, great question. So I just looking for a different paper was published, I think, last year, much better model, same thing. The problem is that if you look carefully what they model, that they need a very fast change of the environment, so that it should be so fast that the children are not, so the parents are not adapted to the environment the children should live in. Additionally to that, most of these models do not include sexual reproduction, so that in reality, in most sexually reproduced species, the long life and these newly acquired traits will rapidly recombine, and you will have also this super new adaptation and long life in one animal very fast. Third, this model doesn't fit very well to the observations we see in nature. So why then, if we speak about eusocial insects, the reproducing caste is living longer? It should other way around, live shorter to ensure variability. Third, so also variability can suffer if you increase genetic drift. So if you increase change of generations, you can lose rare variants that will maybe adaptive at the next corner of evolution. So because variability is very important to preserve. And if we just favor those that are good at this moment, then we can lose it. This is next. Then if you look at animals that didn't evolve for a long time, like sharks or tadpole shrimps, or these solenodons, the mammals that are the same for 66 million years since the Chicxulub impact event, we don't find them very long-lived. So it doesn't seem that they did it good. And I think I have some other arguments to list, but I think this is enough for that time. I can send you the paper in the chat.
I appreciate that.
You
And I find
you
this would
can-
appreciate it. Thank you.
Yeah.
Okay, thanks.
Kent?
I think the last raised hand we have is Kent.
Hey, Peter, good talk. I'm curious about your lab work. How much of it is a bottleneck versus data analysis? And are you frustrated at all about experimental lab bench work? Or is that not really slowing down your research?
No, I'm a virologist by training, and I spent all the pandemics very having fun in BSL-3. So I'm a wet lab researcher. So no, it's totally fascinating. So just I think that it can be very interesting and important for the cellular senescence field. So I think it's not as global, and also I'm testing the model that has been proposed by somebody else. That's why I think I have maybe expressed less enthusiasm about it.
And I think you might have took the question wrong. I didn't know that you actually spend a lot of time at the bench. That's good to know. But are there still a lot of hands-on? So I haven't done full-time molecular biology at the bench myself in 20 years. And I just remember it being incredibly difficult and slow and tedious. And I'm wondering if it's still a pain and if it slows you down, or if other things are greater barriers than the actual experimental techniques and the instruments.
No, everything you do can be frustrating if you don't like them, if they don't work, or you have other more exciting things to do. No, but now I have students to work for me.
So you got grad student slaves, so you're not complaining as much as being.
Oh, well, not
about
slaves, not slaves.
Yeah, I know, but I just is there any experimental scale that you wish you could achieve, but it's beyond your resources? What would you do with limitless lab monkeys or grad students that you can't do now?
It's a very good question. So I would probably invest more in the immune aspects of the thing, because I think what happens with the immune system is key. And I think probably the best route to rejuvenate human body, at least to some extent within our lifetime, is to rejuvenate the immune system. So I would go there. I work on senescent cells because it's easier with the limited resources within a limited time with my expertise. If I would have more, I would go into the more immune aspects.
If you were an investor, would you invest in anti-senolytic therapies, or do you think those are doomed?
Yes, I would.
Okay, so you think it's a good idea to clear out senescent cells, even though they have this really important function?
I think the important functions of senescent cells are happening within a short period of time after they are induced. And this is not only my opinion. I've asked the same question from other experts in the field. They also say so.
Cool.
Next question is Maddie.
Hi, Peter. Thank you. I'm quite curious about other cases of hosts shortening their lifespan in response to infection that might be less severe than the cases of semoparity, but would arise in cases where you might not have eusociality or where the infection might be less severe. In those cases, how would you distinguish the first order consequences of an infection by a pathogen from second order effects of the host response that might be trying to mitigate the impact of the pathogen on inclusive fitness?
Yeah, this is a very good question. Indeed, chronic infections are known to accelerate the aging clocks. For example, HIV or chronic hepatitis are accelerating aging. It's strictly according to our model because if animal understands that it is chronically infected, it may be an indication for it to die sooner. But I think it's now commonly accepted that chronic inflammation even not caused by a pathogen, is bad for lifespan and for health span. So the chronic inflammation causes all the symptoms of aging or accelerate them. In case for infections, no, probably you would like to analyze which immune pathways are induced and then mutagenize these pathways, infect these mice and see if the infection still. But this is a very complex experiment because the mutagenizing these pathways can still affect the infection itself. So it's a experiment. So it is not a trivial thing. But for sure, I think right now, if we just activate with genetic tools, let's say, activate inflammation in animals, then they age faster. This is very well known. So the immune system can indeed accelerate aging. I think this is a fact that, Vadim, would you argue? Vadim dropped.
Sorry, I'm here, but I didn't hear the question. I'm sorry.
So the chronic inflammation accelerates aging, right? So
Yes.
it's this is a dogma right now. So we don't have any more questions. So Vadim, do you want to continue the discussion?
I guess I can. Maybe just a couple of other comments I make. Sorry if I'm critical. I'm not.
This
not
is
critical
very
to you,
welcome.
but it's just a discussion. Yeah, I may be wrong. I may be completely wrong. But to me, I think you mix up not just aging and mortality, but also aging and longevity. So when we consider evolution of aging, we should consider evolution, the transition from non-aging to aging species, and distinguish it from the changes in lifespan, evolution of shorter lifespan and longer lifespan, which may be adaptive. But it doesn't mean that evolution from non-aging to aging state is adaptive. But even if it's adaptive, it doesn't mean to be programmed. Because, for example, like we evolved the brains. Brains have non-renewable, fully differentiated non-renewable cells. Yeah. And some cells lost over time. So by evolving brain, we basically are predisposed to age, right? But it's not adaptive. It's not adaptive aging. We evolve the brain for neuronal function. But as a consequence of that, we age. So it's not like we evolve to age just because we have brain. So the logic is a little bit incorrect to me. And there are several examples like that. Or for example, pleiotropic genes. You say that nobody showed the examples of pleiotropic genes. And that all models like Williams and Medawar models, they're based on entropic factors, which is also not the case.
No, I didn't say that. But wait, sorry for interruption.
Anyway, so I think I said enough.
Okay, so I believe that people who believe that aging is damage, their ideas are coming from analysis of Medawar and Williams from antagonistic pleiotropy. And
for example, I disagree with Medawar and Williams, but I think aging is caused by damage. And there are many people like this. Because to me, every is pleiotropic. Every gene has a function. They do whatever they do, right? But also when they function, they also produce damage. We know that. Mutations, epimutations, again. So byproducts of metabolism, they happen. So they have two traits. One positive function, one negative is damage. So every single gene.
Yeah.
And then you say nobody showed any pleiotropic genes.
Nobody showed. No, so I didn't say that. No, the genes with two functions, one good, one bad, has been shown. But for antagonistic pleiotropy to work is not sufficient. There should be a gene that these two functions cannot be disconnected by evolution. This is not possible because what does it mean? So that, okay, a rat is dying because of some pleiotropic genes, but squirrel lives 5 times longer. So does it mean that squirrel ages because of different set of genes, or the negative effects of these pleiotropic genes that are in rats will somehow compensated in squirrel? And the damage accumulation and antagonistic pleiotropy are specifically bad in explaining this type of arguments. Also with the brain, Vadim, we have evolved like 7 million, our separation with chimps, 7 million years. So in these 7 million years, we first evolved much better brains, maybe a little bit better brains, but also we doubled our lifespan. So there are really no problem to evolve both And then you say that we age because of the brain? No. Typically, animals with big brains live longer. Not all of them, but generally smarter you are, more you tend to key strategy, which is also associated with long lifespan. I think I have missed the first part of your question. What was it?
My example was that you mix up transition from non-aging to aging
Yeah,
versus
yeah.
lifespan. And in your answer, you again mixed up those things.
Yeah. So this is a complex discussion, and because indeed in my models I mostly focus on lifespan, because this is a parameter in the epidemiological model. Aging is more like a mechanistic core of this lifespan regulation, and I'm just not ready to answer this question because I need to a little bit tilt my mindset to understand better, and I'm afraid that if I will start to speak now, it will be rubbish. I need to think. If you can write me, formalize the question, I will try to answer it. Yeah, I would be very grateful if you can do it.
I think that's all we have for today. Thank you so much to everyone for your great questions. Thank you, Professor Peter, for coming on to the series, and hope everyone has a great evening or morning or afternoon, wherever you are in the world. And have a great January, and we'll see you next month. Bye-bye.